Hi everyone. Starting from today and the next three lectures, we'll discuss molecular biology. We have learned a lot about chromosomes in previous lectures, and starting from this lecture, I'll introduce chromatin. This is another form of DNA. DNA was first isolated by Friedrich Meissner, a Swiss physician and biologist. Friedrich was interested in studying the chemistry of cell nucleus. At that time, it was known that the neutrophils were one of the main components in pus and could be obtained from bandages in hospital. So Friedrich developed a method to isolate nuclei from the pus. He called the contents in nuclei as nuclein and determined that the nuclein was made up of hydrogen, oxygen, nitrogen, and phosphorus. Now we know that nuclein is actually DNA. His discovery played an important part in the identification of nucleic acids as the carriers of inheritance. Research in the first few decades of the 20th century identified chromosomes as carriers of genetic information, but it was unknown which chemical components was the genetic material. There were two important experiments establishing the role of DNA as the carrier of genetic information. So one is the Griffiths experiment reported in 1928 was the first experiment suggesting that bacteria were capable of transferring genetic information through a process known as transformation. And the second experiment performed by Hershey and Chase were a series of experiments conducted in 1952. And then that experiment helped to confirm DNA is the genetic material. I'll briefly go through those two experiments. So this is the first experiment performed by Griffiths. So Griffiths used two pneumonia strains. So first one is the S strain. So S strain is virulent and it can cause death in mice. In contrast to S strain, there's an R strain. The R strain is non-virulent and does not really kill the mice. But then the virulent S strain is heated and being killed by this heating, and then the mouse is fine because the S strain already dead. But interestingly, when Griffiths he mixed up the living R strain, which is not virulent, with the dead S strain, and then what happened? The mice actually died. So based on this observation, Griffiths gave the transforming principle, which states that something was getting transferred from heat-killed S strain to this live R strain, and that made the R strain virulent and killed the mice. In the late 1940s and early 1950s, scientists began to study viruses that attack and kill bacteria. These viruses were named bacteriophages or simply phages. Phages consist only of DNA and a protein coat, making them ideal candidates to help identifying which of these molecules carry genetic information. So this picture showing here is an electron microscope picture that bacteriophages attacking a bacterial cell. So you can see that phage DNA, they enters the bacterium, whereas the protein coat, they stays outside. Because phages containing only DNA and proteins, Hershey Chase experiments used a phage as the model or tool to study if DNA or proteins are genetic materials. At that time, it had been known that phage proteins contain sulfur but not phosphorus. And the phage DNA contains phosphorus, but not sulfur. So Hershey and Chase designed 2 experiments to test whether DNA or protein contain the genetic information needed to direct the replication of new phage particles. Here shows one experiment. So they infected the bacterial cells with viral articles whose protein was labeled with radioactive sulfur or S35. So now you probably already know why protein can be labeled with S35, right? Because you learned about chemistry. This is because we know protein are composed of amino acids, right? So amino acids have the same backbone structure, but their side chains are different. So we have 2 amino acids, namely methionine and cysteine shown here. Both of them contain the sulfur in their side chains. And then the isotope labeling process means replacing this non-radioactive sulfur with a radioactive sulfur, this S35, so that the radioactivity can be detected using, later using some machine. So in this experiment, it was found that the radioactivity remains outside of bacteria cells, after the infection, meaning the phage proteins do not enter target cells, stays outside. But in a second experiment, this time the viral DNA was labeled. This time radioactive phosphorus called P32 was used for labeling. But why? Because DNA has phosphate group, right? So we'll get that in a while. So they found that the radioactive labeled DNA this time enters the bacterial cell and directly the synthesis of new viral particles. So this provided more evidence that DNA, but not protein, is the genetic material. Okay, since DNA was identified as a genetic materials, scientists have spent much efforts to understand the chemistry and the structure of DNA. Before I go to the detail, I would like to review some basic chemistry. So here shows 2 types of chemical bonds involved in the DNA structure formation. So the first one show in A, this is a covalent bonds, all right? So covalent bonds are shown as solid lines that connect atoms, right? Depending on the degree of electron sharing, they can be 1 or 2 here. They have covalent bonds between those atoms. So once formed, covalent bonds are stable and are broken only in chemical reaction, so very, very strong. But then the second bond shown in B, this is a hydrogen bonds, okay? Hydrogen bonds are usually represented as dotted lines, showing the here. So this one is shown as water molecules form hydrogen bonds with adjacent water molecules, all right? So we know that water is H2O here. So these hydrogen bonds are very weak interactions and easy to broken by heat or molecular tumbling, and then they can reformed with other water molecules. DNA is composed of simpler monomeric units. We call the nucleotides, okay? Each nucleotide is composed of one of 4 nitrogen-containing nucleobases. One is the cytosine C, guanine G, adenine A, or thymine T. So a sugar called a deoxyribose and a phosphate group. So that's the composition. Similar to DNA, RNA is also made up of nucleotides. However, the composition of the bases and sugars are different between DNA and RNA. All right, so DNA Here you have a ATCG, GC, and then in RNA, it does not have a T anymore. In RNA, it has a uracil instead of a thymine. So, but then for the sugars, and then in the DNA, you have this deoxyribose, but then in RNA, it has the ribose. So this group is different. Pay attention to that. And then they both have the phosphate group. This slide shows polynucleotide chain when nucleotides are joined together by covalent bonds. Remember, they are joined by covalent bonds, okay? Polynucleotides are polar molecules with a 5' end at the phosphate group, and then a 3' end at the sugar group, okay? An RNA polynucleotide is shown on this on here, and the DNA polypeptide is shown on the right. All right. Now let's watch a short movie on discovery of a DNA structure.
We are in 1953 in Cambridge, a city where scientists are racking their brains to corner what was known as
We have discovered the molecule of life.
Yes, that one. Okay, let's rewind a little. Several researchers over many years discovered that in the nucleus of our cells there was a substance called DNA, which contained all our genetic information, and it was made of 4 compounds: adenine, thymine, cytosine, and guanine. How was it possible that the whole book of our life was written with just 4 letters? Our celebrating friends James Watson and Francis Crick thought that maybe the answer was in the structure of DNA, and they were trying to solve this 4-pieced puzzle that was driving all the scientific community crazy. They had 2 clues. The first one was a discovery made by Erwin Chargaff, who found that in all organisms he tested, the relative amounts of adenine and thymine were equal, and the same happened with guanine and cytosine. The second clue came from Rosalind Franklin, an experienced chemist who generated DNA crystals and shot X-rays through them to study their structure. The X-ray picture showed a repeated pattern, which meant it was something similar to a helix. Watson and Crick tried all the different possible unions between A and T and G and C, and one was perfect. 2 bonds between A and T and 3 bonds between C and G would fit in a helix. A double helix indeed. Also, the 4 units structured this way could code a huge amount of data, as a dot and a dash can compose a whole alphabet in Morse code. On top of these discoveries, specific pairing suggested the copying mechanism for DNA, for the production of new cells and new life. 10 years after the celebration in the pub, Watson, Crick and Maurice Wilkins, the boss of Franklin, received the Nobel Prize for this discovery, and the DNA revolution spread across every biology laboratory in the world.
So as a summary, DNA is a double helix and has twisted ladder conformation. The 2 polynucleotide strands are the rails of the ladder. The 2 strands are held by hydrogen bonds forming between the bases, similar to the rungs of a ladder. The base pairing is highly specific. A can only pair with T, and G can only pair with C. I have mentioned that DNA has 2 ends, 5' end that bears a phosphate, and 3' that has a hydroxy group. We'll use these directions in a lot in the future lectures. But remember, the 2 polynucleotide chains in DNA run in opposite directions. So the left strand runs from 5' end to 3' end, and the right will be 3' end to 5' end. So the base sequences in each strand are complementary. An A in one strand pairs with a T in the other strand, and a C in one strand is paired with a G in the opposite strand. They're always like that. Another important point to remember is that 2 hydrogen bonds form between T and A, showing here, T and A, you can see 2 hydrogen bonds versus 3 hydrogen bonds between C and G. So you can tell that DNA strands are held by hydrogen bonds. Because hydrogen bonds are non-covalent, meaning they are breakable, particularly when DNA molecules are heated. So do you think if TA is more difficult to break or CG? Yes, CG has 3 hydrogen bonds are more difficult to break. So now let's do a question. So here I have 2 double-strand DNA fragments, 1 or 2. So both are composed with 6 nucleotides. In your opinion, which DNA fragment is more resistant to heat? Here you can pause it. The correct answer is the fragment on the right, this one, okay? So this is because it has 4 CG pairs, but the one showing here, they only have 1 CG pair, okay? The Watson-Crick DNA model also suggested that genetic information is stored in the bases of the DNA. Now we know that genome is a complete set of genetic information, and the size of the genome is species specific. And because DNA is the genetic material, any changes to the DNA sequence, like mutations, can result in an altered phenotype or even disease. This table lists the genome sizes of some organisms. You can see bacterium has this is as a prokaryotic organism, the genome size is rather small in comparison with eukaryotic organisms. However, it is interesting to note that tobacco plant showing here has bigger genome than our human being. Therefore, genome size is not always correlated with genome complexity. So we'll learn the reason in our future lectures. Now I would like to spend a few minutes to remind you about RNA structure and function. RNA is a single-stranded polynucleotide chain. The same as DNA, RNA molecule also contain 3 components: nitrogen-containing bases, sugar, and a phosphate group. The difference is that it has a ribose sugar instead of a deoxyribose, and RNA has a uracil in place of thymine. Same as DNA, the RNA polynucleotide chains are joined by covalent bonds. But different from DNA, which forms a double-stranded helix structure, RNA is a single-stranded. Therefore, it has to fold upon itself. So with the folds stabilized by short areas of complementary base pairing, showing, for example, showing here, this is inside the molecule. This is going to form a 3-dimensional structure. Okay, so based on the central dogma, we learned that RNA mainly function in transfer of genetic information from the nucleus to the cytoplasm and in the protein synthesis. This table summarizes the molecular differences between DNA and RNA. We have talked about it, so I will not repeat, but I would like to ask you a question. So what is the electric charge of DNA? Is that positive or negative? Yes, it's negative. But why? Because the phosphate group is a component of DNA. Okay, good. Then how about the RNA? So RNA is also negatively charged because RNA also have phosphate group. So when we have the exam, I do not have to ask what are the chemical components in DNA or RNA. I could ask differently, such like in this way. So if you understand the concept, you could easily choose the correct answer. Okay, so now I'll move to gene, and we'll introduce the gene structure. Okay, so in our first lecture, we learned that gene is a region of DNA that encodes a function. But how can we tell where gene starts, where gene ends? Does gene have specific structure? If DNA forms nucleosomes with histones, how do they organize? Here comes one of the most important slides for this course. This is a central dogma, and we're talking about phenotypes, genotypes, and we now know that DNA is basically genotype, and protein, the gene product, is basically phenotype. Okay, so let's first take a look at the gene structure. For each gene, you almost always find an enhancer, a promoter, exon, and introns. When I was a college student, there was no concept of enhancer, but now we know for gene expression, enhancer is probably more important than promoter. Okay, in genetics, a promoter is a sequence of DNA to which proteins bind that initiate the transcription of a single RNA from the DNA downstream of it. So it is right upstream of the gene. Then enhancers are regulatory elements that actively promote transcription over large distances. So enhancers are usually far away from the gene. They have this dotted line showing this distance. Exon is the portion of the gene that encodes for amino acids, and introns are nucleotide sequences in DNA that do not directly code for proteins, and they are removed during splicing. These concepts should have been introduced in your previous intro bio or general genetics before you taking this course, right? But then transcription results in production of pre-mRNA or pre-messenger RNA. So based on the sequence, we name the transcription starting site here shows as TSS and the transcription termination site as TTS. Then the splicing event removes the intron in pre-mRNA, right? And when mature mRNA is transferred from nucleus to cytoplasm, translation occurs and makes protein. You may notice that I color exon 1 and exon 3 as different colors. This is because during the translation, if you remember, the translation starts at a codon with ATG and stop at stop codons. So we, for the sequence upstream of ATG shown this blue color, we named as 5' UTR, 5' untranslated region. Similarly, for the sequence downstream of translation stop codon, we call it 3' untranslated region or 3' UTR. So I hope you can understand everything on these slides because this is going to be important for get a really good score in your exam. So when you see this picture next time, I wish you could tell which structure is what, right? So overall, from TSS to TTS, so this DNA fragment we call a gene body, all right? So now you can tell that gene body is the transcriptional region of a gene, and it contains both introns and exons, all right? But in contrast, enhancers and promoters are characterized as regulatory regions, as they are DNA sequences that are capable of increasing or decreasing the expression of specific genes. What I have talked so far are all at a DNA level. The organization of DNA into chromatin is important, as each of our cells have about 2 meters of DNA, and they need to be fit inside a tiny nucleus with a 5 micrometer diameter. So during cell cycle, chromatin and chromosomes need to be uncoiled and recoiled. For a long time, scientists thought that the chromatin is disordered during interphase, like a bowl of spaghetti. Now we know that in most eukaryotes, chromosomes largely occupy defined regions of the nucleus, even in the interphase. So we call this as chromosome territories. Chromosome territory means that each chromosome occupies a spatially limited domain, chromosome territory. So this picture shows a human chromosome territories during the prometaphase in a fibroblast cell. In the interphase, although the chromosome territories are not so obvious like this picture showed, but the concept still holds. The chromosome territories are linked to function, meaning gene expression is regulated by chromatin arrangement. Let's see how chromosomal territories affect the gene expression. In this figure, if we look further into each chromosome territories, as shown in B here, this is amplified basically, we can find structures named topologically associated domain, or abbreviated as TAD, okay? TAD is a self-interacting genomic region, meaning that DNA sequences within a TAD, they physically interact with each other more frequently than with sequences outside the TAD. For example, if you consider the University of Houston as one chromosomal territory, and our class can be one of the TADs in this territory. So although the size of TAD varies, its average size of TAD is about 1 millibase pair. So in the TAD compartments that are located close to nuclear membrane, shown here as the pink color, the repressed TAD compartments are enriched. Here, the repression really means gene transcription level is low. So this is inconsistent with what we learned before, because we learned that heterochromatin is enriched in perinuclear membrane region, and we now know that the heterochromatin contains very few encoding genes. So transcriptionally, it's kind of silenced or repressed. But there are also active TADs, these compartments showing here. So these TAD regions contain genes that have high levels of transcription. So there are certain DNA sequences that set a boundary for each TAD, showing this red colored boundary here. So the boundary proteins such as CTCF, that's a protein's name, they can specifically bind to the boundary sequences and establish the territory. So within this TAD showing here, so the regulatory elements can physically interact with genes, such as those could be enhancer, could interact with the gene showing here, or over here, there's another enhancer interact with the gene, and then they will activate the gene transcription. So here is the summary of TADs. This is a relatively new field, and it was first discovered in 2012. And up to now, scientists still have very limited understanding about TAD. Now let's take a look at the basic unit of packaged DNA, or nucleosomes. DNA coils around the outside of a histone cluster to form a nucleosome. So this is the first level of DNA compaction in chromosomal structure. So the picture showing on this left is a crystal structure of the nucleosome, and the double-stranded DNA are shown as in this gray color. The picture on the right, this is a cartoon that clearly shows nucleosome is made of core histones in the middle and the DNA wrapping around. So under the electron microscope, the chromatin shown as a bead on a string structure with multiple nucleosomes linking together. Okay, the nucleosome core particles contain approximately 146 base pair of DNA wrapped in about 1.67 left-handed superhelical turns around the histone octamer. Okay, so octamer, that means it has 8 molecules, right? So basically it contains 2 copies of each of the core histones named H2A, 2B, histone H3, and histone H4. So another histone molecule named histone H1 helps to lock up the core particles. So histone H1 is not a core histone, remember. The core histone only these 4, okay? The core particles are connected by stretches of linker DNA, which varies in length from 8 to 114 nucleotide pairs. All core histones have a long N-terminal tails that extended from the core particles, but it did not show here. So because these tails do not form a stable structure, so they cannot be seen clearly under the microscope. Therefore, they are not included in these pictures shown today. However, these histone tails play very important roles in gene regulation. We will discuss those in our epigenetics lecture in the future. Okay, so let's play some, practice some questions together. All right, these are the true or false questions. So the first one, DNA contains 2 identical polynucleotide chains. Is this correct? So the answer is actually false. Apparently, the polynucleotide chains are not identical, as nucleotides have to match to each other. It's complementary. So A goes with T and C goes with G. So this is false. Okay, second, this is the interface nucleus has an internal structure in which each chromosome as the form of chromatin occupies a distinct region called a chromosome territory. Is this true or false? So this is true. Chromosome territory also includes the chromatin structure in interface. So please do not get confused by the word of chromosome here, okay? Okay, next one. The process of transferring genetic information between bacterial cells is accomplished with a transformation factor called DNA. This is true or false? Okay, so the answer is true. Actually, Griffith's experiment proved this point, right? And the false one, because females carry 2 X chromosomes and males carry 1 chromosome, females have higher levels of OPRA encoded by genes on the X chromosome. This is from the last lecture. So the answer is a false. Why? Because X inactivation, only 1 X chromosome is transcribed. So the gene product from X chromosome is comparable between females and the males, right? Okay, that's next. The brain and nervous system of a developing embryo can only be damaged during the very early stages of development. True or false? This is apparently false, because we learned that the toxic effect on brain and nervous system spans through the entire phase of pregnancy, right? Okay, the last one. Phenotypic sex is determined at the moment of fertilization. True or false? So the answer is false, because we learned that genotypic sex is determined at the moment of fertilization, but not the phenotypic sex. So phenotypic sex is the last stage of sex determination. See next. The basic building block of DNA and RNA is a what? Okay, you can pause if you want to See, but this answer is the D. This is actually quite obvious, very straightforward question. Second, histones are the, you can pause here to think and then. So the answer is A. So chromatin is made of DIN and histone. So all the other answers are trying to confuse you actually. So next. So bacteriophages are viruses that what? You can pause here again. Okay, the answer is E. We learned that this from the Hershey and Chase's experiments, right? So all other choices are incorrect. Next one. So nucleosomes are bead-like structures. And then what? Again, you can pause here to think. So the answer is a C. If you understand the components of nucleosomes, you won't get this wrong. So B is probably a tricky answer, but but here it asks about nucleosomes, not histones. So only C is correct. Okay. Another one, the undifferentiated the genitalia of an early human embryo develops into male genital under the influence of what? So this should be pretty easy. So the answer is D. Male reproductive organs need to have androgens to develop, right? So the male gonads secrete testosterone and AMH for this development. So those, both of them are a type of androgens. Okay, a blastocyst. So you need to know what a blastocyst means, right? So this problem, the answer is C. So a blastocyst is an early embryo when implanted into the endometrium. So B is the ovary that release the egg, and the D is a trophoblast, right? So E really means placenta, it's the placenta's function. So the only correct answer is C. Okay, so this is the last slide showing you teratogen affects what more than any other part of the fetal body. So we have played this already. So that's the central nervous system. The answer is A. Okay, next. The most important events in human development occur during the what? Different phases. Okay, the correct answer is C. So organ formation occurs during the first trimester. Although all the events during pregnancy are important, but without a correct organ formation, miscarriage will occur. So therefore C is the only correct answer, okay? So this is also the earliest developmental period. All right, that's all for today.